Hello everybody! In this video lecture, we will cover mechanism of evolution and speciation. So this is chapter 11, part 3. So what is the modern synthesis? So modern synthesis is the fusion of genetics with evolutionary biology. And we will discuss it in this lecture, but before we begin, let's give definition of a gene pool. Gene pool is a total number of alleles of every individual in a population. So on this diagram below, we can see representation of the gene pool of domestic dogs. So if you imagine that we will have imaginary pool with all these alleles that code for all different characteristics, drop them in this pool, and this will be a gene pool of this particular species or this particular population. So if we look at small population, for example, this is the population that we want to study, right? And this population is really very, very small. It has only 10 rabbits. And you can see some rabbits are brown and some rabbits are white. And we know that brown fur color is dominant and white fur color is recessive. And because recessive trait, for recessive trait to be expressed, we need homozygous recessive genotype. So it's little a, little a, little a, little a. For brown rabbits, however, it can be homozygous dominant or heterozygous. However, there is a formula to calculate how many individuals will have big A genotype and how many individuals will have heterozygous big A, little a, right? But we're not discussing the formula, but let's imagine that we already know how many dominant alleles and how many recessive alleles we have in this particular small population. And we can represent frequency of dominant allele with P and frequency of recessive allele with Q. Right? So in the first generation, P equals 0.5 and Q equals 0.5. Because if you count number of capital letters and number of lowercase letter, it's going to be 50-50. Right? So that's our original population. Now, if we are evolutionary biologists and we study this population in nature, and then we come back to this population maybe a couple, years later, or maybe even next year, and we look at the second generation, and what happened over here, actually now we have only one white rabbit. So we can calculate frequency of dominant and recessive allele again, and you can see over here in the second generation, frequency of P is 0.7 and Q is 0.3. Right, then we again leave this population for a while, come back next year, and now looking at the population, we see that frequency of P is 1 and frequency of Q is 0. So what's going on over here? Frequencies of alleles are changing over time, right? So when the relative frequency of alleles changes over a time of generation, evolution is occurring on its smallest scale, right? So using modern theory of synthesis, we can redefine the evolution. So today, this is the definition we use for process of evolution. Evolution is changes in the population allele frequencies over time, right? So, if population frequency are changing, population is changing. As a result, evolution is occurring, right? So that's our definition, modern definition for process of evolution. So we already mentioned that alleles can be symbolized P for frequency of dominant allele, Q for the frequency of recessive alleles, and P plus Q always equals 1. and Hartley-Weinberg formula allow us to calculate the frequencies of the allele and frequency of genotypes, right, based on a phenotype that we can actually see. So if you can make observation about phenotype, you can use this formula to calculate frequency of alleles, right? And then if you can calculate frequencies of a population, you can see are those frequencies are changing or not, and if they change, generation over generation, the population is evolving. So how can we tell if a population is evolving? A non-evolving population is in genetic equilibrium, also known as Hardy-Weinberg equilibrium, meaning the population gene pool is constant over time, right? So from a genetic perspective, evolution can be defined as generation to generation change in the population's frequencies of alleles, sometimes called microevolution. And today we even, say that there is no microevolution. Microevolution, macroevolution, they are very similar processes. Now, if we want to change frequencies of allele, how to do it, right? What are factors that can lead to evolution? Or what are the factors that can lead to change in allele frequencies, right? So imagine you have a big population and it lives, hundreds and thousands and hundreds of thousand years, and really it's kind of seems like it stays the same. Now, what can be done for this population to change? And remember that population is changing, that means population is evolving, because evolution is a change. So this is 5 factors that can lead to evolution. And to remember them, we're going to use your own hand, right? The small finger, little finger, is for small population. So when population became smaller, then it can lead to changes in their allele frequencies. And when population becomes smaller, we call it genetic drift. Now this ring finger should remind you sexual selection. So sexual selection can lead to evolution. Middle finger starts with M, and it should remind you about mutations. So mutation changes alleles, and as a result, frequencies are changing. This finger over here, that's the finger you use to point to the object, and this should remind us about gene flow. When some individuals leave population or enter the population, that also can change allele frequencies. And the big finger, natural selection. So nature votes finger up for traits that are useful for survival, or thumb down for traits that doesn't allow or reduce population chances of population to survive. So small population, sexual selection, mutation, gene flow, and natural selection, all of these factors can lead to evolution. So we're going to talk about small population first, and when population became small, we call it genetic drift. So genetic drift is a change in the gene pool of a small population due to chance. So you have a big population, right? And then something happened, maybe, I don't know, frost or earthquake or volcanic eruption. Something that kills lots of the individuals within the population, or maybe it can separate larger population into smaller population, right? But something that happened random, that's genetic drift. And there is 2 types of genetic drift, bottleneck effect and founder effect. So bottleneck effect. Bottleneck effect is genetic drift that results from event that drastically reduces the size of the population, right? And results in a large portion of the genome suddenly being wiped out. And they call it bottleneck effect because it's like a neck of a bottle. So if this bottle represents the original population, and you can see we have all these different alleles. It's representation, don't forget about that, right? But we have some green, some pink, some red. But when population reduces in size, only few individuals survive. So that's our surviving population. And look over here, we lost this red allele, right? So this population now is different from original population, and that means allele frequency changes. That population is evolving. Is it good for population to go through bottleneck effect? No, it's not good. It's always a bad thing to lose genetic diversity, but nevertheless, population is different, population is changing. That means it's evolving. So here's an example of bottleneck effect. Cheetahs appears to have experienced at least 2 genetic bottlenecks during the last ice age about 10,000 years ago and during the 800s when farmers hunted the animals to near extinction. So you see in both examples, population became very small. So we saw little variability. Cheetahs today have a reduced capacity to adapt to environmental changes. Another example of genetic drift is founder effect. So the founder effect is likely when a few individuals colonize an isolated habitat. The founder effect explains the relatively high frequency of certain inherited disorders in some small human populations as well. So here again, representation of founder effect. So that's our original population of butterflies, and we have higher variability over here, brown and black and white butterflies, and only some of them colonize a new area, and some alleles might be lost, or they can be reduced in numbers. So this population is different from original population. Population is changing, population is evolving. So here's a real example. In 1775, catastrophic typhoon swept through the island, killing 90% of the inhabitants and leaving approximately 20 people. It is believed that one of the survivors, namely, you guys try to read it yourself, I'm gonna leave it for you, but that guy was a ruler at that time, and he was a carrier for complete Achromatopsia. Yeah, Achromatopsia. So that's a rare genetic disorder that results in total color blindness. Color blindness is not that rare, but usually people just cannot distinguish between 2 different colors, but being totally colorblind, that's very rare. So if you look at these pictures over here, that's a normal vision, and this is how a person with this disorder would see the same area. So it's like old black and white TV, right? So now look, if many people died, only 20 people left, right? That's a very small population, right? And the guy who survived was a ruler at that time. So I don't know about other guys, but that guy probably had a wife. And even if his wife was killed being a ruler, I'm sure he found a female out of these 20 people who would give him kids, right? And that means if maybe, you know, some guys with normal vision over there, they just couldn't pass their normal genes. But this ruler guy, he passed this gene for achromatopsia to his kids. So by generation 6, the percentage of people with color blindness disorder rose to approximately 10%. Right? So that was a small population, and in this population, the own changes within gene pool made it a little bit different from the rest of the people. So that's called founder effect. So gene flow is another mechanism for evolution. It is separate from genetic drift. It's genetic exchange with another population and may result in the gain or loss of alleles. And gene flow tends to reduce genetic differences between population. So over here, our population 1 and population 2, and they used to be different, but because of the immigration of white geese here, right, this population is changing. That means this population is evolving, but at the same time, we reduce differences between them. Gene flow is absolutely amazingly represented among human population because like 300 years ago, we were so much more isolated from one another. And now we're moving through country to country and we take our genes with us. How many of us came to United States as immigrants? I am one of them, right? Not only I came to this beautiful country, I also brought my genes here with me, and I brought my daughter here, and my daughter now, she has her own kids, right? So that's my own family, and maybe many of your families. Example of this gene flow that changing the populations, and the population are changing, population are evolving. Remember, another mechanism for evolution is mutation. So mutations are any changes in an organismal DNA. Alone, one mutation or even several mutations, they do not have much effect on a large population, but can have significant cumulative effect on a population. So here's an example of mutation in human population, sickle cell anemia. And we all have a DNA, right? In one gene, we have this gene that code information for protein synthesis, and this protein is hemoglobin, right? So inside DNA, instruction how to make hemoglobin, and if it's a normal gene, then your red blood cells look like this, like a donut. And some people have a mutation in the DNA, sickle cell mutation, and it's result in a sickle cell red blood cells, right? So that, here's the cases. And what is interesting about these numbers is that sickle cell disease found 1 in 500 of African American births and 1 in 36,000 in Hispanic birth, for example. So how can we explain this difference? There is actually good explanation for those numbers. So what scientists figure out that the sickle cell anemia is actually present in a high frequencies in the area of Africa where malaria is common. So malaria is caused by Plasmodium, and this is a protist, so it's a little microscopic organism. Sickle cell anemia, however, right, it's a mutation in the DNA that changes the shape of red blood cells. So how can they be related, right? So researchers found that the sickle cell gene is especially prevalent in areas of Africa hard hit by malaria. In some regions, as much as 40% of the population carries at least one sickle cell gene. So if you look at this Punnett square, and sickle cell allele is a recessive allele. So for a person to have sickle cell disease, they must have little a, right? Homozygous recessive. So this person has sickle cell anemia. Unfortunately, this person can die, right, from the hypoxia, right? So not enough oxygen in their bodies. This person is perfectly fine, no sickle cell anemia. But look at these carriers. These carriers, they don't have sickle cell anemia, at least not at a very extreme degree, right? So they don't die from sickle cell anemia because they still have one normal gene, right? But what researchers noticed, these people resistant to malaria. So now imagine what happened. We have population of people and it's hit by malaria. So people dying and dying and dying from malaria, right? And some people die very young and they don't have chance even to have their own kids and pass their genes. And then something happened. Mutation happened, right? And mutation happened that gave these people some resistance to malaria. Now they survive malaria and they live longer. They have more kids and they passing their genes. Unfortunately, this solution comes with, another disease, with sickle cell allele. It was not the best solution, but evolution doesn't work this way. You cannot choose the right weapon fight against the malaria. This was available one, right? And these people survived, and that's why we can see this high percentage of sickle cell allele in Americans of African descent, right? Because that mutation gave them some advantage and allows them to survive malaria. Okay, so it turns out that in this area, sickle cell carriers have been naturally selected because the trait confers some resistance to malaria. Their red blood cells containing some abnormal hemoglobin tend to sickle when they are infected with the malaria parasites, and those infected cells flow through the spleen, which cuts them out because of their sickle shape, and the parasites are eliminated along with them, right? So these people have some sickle cell red blood cells, right? But they still have normal cells as well because they only carriers, right? And this sickle cell, they just removed along with the parasites from the body. So here's another mutation. It's called Delta 32 mutation. And well, this is the case that case that was not researched that well yet, but on this picture you can see a special human cells that called T helper cells. These cells help you with the immunity, and this is a virus, and it's well known, HIV virus, right? So for HIV virus to infect our cells, the cells, T helper cells, must have a special receptors. But people with this mutation, they don't have a receptor, right? So you see, there is no this green protein over here, so HIV virus cannot affect their cells. So they have immunity to HIV, right? So, and this is only happen in people of European descent. So is it somehow similar to sickle cell anemia or not? Scientists believe that it might be. Scientists believe that Delta 32 mutation happened between 1347, 1350, somewhere in that area, when about 40% of Europeans were killed by bubonic plague. So at that time, some people survived, possibly because they had this mutation, and now the same mutation protects them from HIV virus. But again, we don't know for sure yet. It's just some interesting case to do research about it. But that's how mutations can change human populations. So here's another interesting example. This is called Macan children. So that's little tribes in Southeast Asia. And children, well, how most people live over there, they collect food from the sea floor without using any visual aids. And investigators shown that the Marken children actually they can do something underwater. They can constrict their pupil, and pupil is that red spot, right? It's an opening. It's a, oh, I'm sorry, black opening in your eye, right? It's an opening, and we cannot dilate or constrict it. We cannot regulate it, but these people can. When they constrict their pupil, they can see clearly underwater. That's very interesting. So that's some adaptations that they have because their well-being, their life depend on this ability to see clear underwater, right? It gives them food, it gives them money, it gives them opportunity to get married, right, and have kids, because now they have food, they have money, they can afford, more kids, and they passing those genes, right, that allows them to constrict the pupil to, their kids. This is completely absent in European children, right? So that's also mutation, and this is adaptation that allow people to, survive better and pass their alleles to the next generations. Okay, another mechanism for evolution is sexual selection. It is a form of natural selection in which individuals with certain traits are more likely than other individuals to obtain mates. And there is a different type of sexual selection. It can be male competition over here or female choice. And if you look at this picture here, like this peacock shows his beautiful, tail, and well, he wants female to choose him for breeding, right? And by showing this beautiful tail, he tells her, look, I am healthy. I can find good food because, if I'm not healthy, I cannot grow a tail like this. I'm good at fighting. I'm good at finding food. I'm healthy. I have good genes. Breed with me, and our offspring will inherit my genes. That's what sexual selection is about. And of course, natural selection. So we're gonna look closely at natural selection. So of all causes of microevolution, only natural selection promotes adaptation. So the core difference between natural selection and genetic drift is the cause. Both are methods of genetic change in a population. However, the genetic drift happens randomly, while the other is a direct response to and environmental change. Right, so if you look over here, we have mutation. So mutation can lead to population. Gene flow can lead to population, right? And here's our genetic drift, and here natural selection. And of course, this is very simple, but that gives us an idea. So if you have population of beetles, and we have green and brown one, and then something happened, right? Like this guy just stepped on this population, and by chance he killed green beetles, right? So now we have all brown beetles, but not because they better adapted, they just was lucky at that particular moment, right? That's genetic drift. So look, population is changing. Remember, population is changing, it's evolving, but why it's changing? It's completely random. So that's genetic drift. On natural selection, on the other side, we also begin with a population with green beetles and brown beetles, but here our selective predator. And predator is not gonna waste time and energy to look for prey. Predator gonna get what is more available, and maybe it can see green beetles better. So green beetles are not well suited to this environment. It's better to be brown because this lizard cannot see brown beetles. So lizard gonna eat green beetles and population changing, right? The brown allele increase in numbers. So here population is changing as the response to this selective pressure. Now, there are three types of natural selection, and the first type is disruptive selection. And disruptive selection is changes in population genetics in which extreme values for a trait are favored over intermediate values. So an example giving to you is actually salmon, right? So relative size of male salmon. So that's original population shown here in gray. And, you know, some fish is small, some fish is large, but most of the fish right here in a medium size. But what if being medium size is not good, right? Why? Because the large fish over here, it can really, outcome the medium size for territory, right? So that's a big fish. So that's gonna, it will out-compete for a territory, right? And being small also not bad because they can sneak in and fertilize eggs. So look, that's especially male, right? If you're small, you sneak in and fertilize, so you're passing your genes. If you're large, you just overcompete, right? And you fertilize an eggs. So it's just not good to be medium size in this particular example, right? Then population will change. We will see more of smaller fish and more of larger fish. So that's our extreme values, and they favored over intermediate. So we will have very few individuals medium size. So that's disruptive selection. Stabilizing selection is opposite to disruptive selection. It favors the intermediate variants. An example is birth weight of a human baby, and here it's shown in pounds, right? So we'll imagine maybe 500 years ago, right? So human babies are born, some maybe 3-4 pounds, some are born 11-12 pounds. But when baby born that small, and remember, I'm talking about 500 years ago, right? When medicine was not that advanced yet. So these babies were very vulnerable to complications, and they maybe died, right? Or if baby born that big, that's also complication. Plus, even mother can die, right? From complication from the labor. So what babies survived? Right here, right in the middle, 7, 8, 9 pounds. And that's why this is the most common birth weight for humans today, right? So that's our stabilizing selection. Directional selection, and I'm reminding you, we're talking about natural selection, right? We talk about disruptive selection, stabilizing selection, and this one, the last mode, directional selection. So directional selection occurs when one extreme favored over another extreme. As example, over here, selection in turkeys on a poultry farm. This is artificial selection. But again, imagine we have turkeys, and this shows the size of the breast muscles. So right here, big breast muscles. So some probably turkeys have a smaller, some are larger breast muscles, but most were somewhere in the middle. But if farmers allow only birds with a large muscle to reproduce, right? Only they select for this large breast muscle. Then what? Then we see the population is changing. We have more and more turkeys with a large breast muscle, right? So turkeys on poultry farm can no longer even mate naturally. Their breast muscles have become so large that artificial insemination is required. But population is changing. We choose one extreme, large, over another extreme, small. So on this diagram, we can see all three modes of natural selection: directional, disruptive, stabilizing. And let's say if we take population of mice here, depends where this population live. we can see all these different type of selection. Now, mechanism of speciation. Diversity of life evolves through speciation, the process in which one species split into two or more species. So how it happens that we have so many species, right? So that's what evolution explains us, that one species split into two, then each one splits into two, then they can split into 3 and 4 and so on. So the process of splitting of one species into more species. But now, what is species? Species, I think, right? So species is group of organisms. They can breed and produce fertile offspring. So an example over here, horse and donkey. Well, horse and donkey can breed and produce mule, right? So they can breed, but mule is not fertile. Mule cannot breed with donkey, mule cannot breed with horse, and mule cannot breed with another mule. So if you want to get a mule, you again need to start with horse and donkey. So that tells us that horse and donkey are not the same species, right? However, they're really closely related. So species must not only breed, but also produce fertile offsprings. Well, how about bacteria then? Well, the definition of a species as a group of interbreeding individuals cannot be easily applied to organisms that reproduce only or mainly asexually. Let's say bacteria. We cannot use this definition for bacteria. Bacteria just splits into two through binary fission, right? So here is a better definition for species. Species is a group of organisms sharing a common evolutionary ancestry and having a common gene pool. So a key event in the potential origin of a species occurs when a population is somehow cut off from other population of the parent species. And species can form by Allopatric Speciation and Sympatric Speciation. Patric means land, and allo means different, and sim means the same. So look over here. Imagine that that's a regional population. So they can interbreed. So they have common gene pool. They exchange different alleles through reproduction. And now something happened. Well, this is a range of mountains growing between them and separated one population into two populations and separated them in a way that they cannot interbreed anymore. And if this barrier stays for a long time, what might happen that now we might have two different species. So if they come back together and they cannot interbreed, then we have two different species. So that would be allopatric speciation. Sympatric speciation happens without geographic isolation, right? And how it happens will be explained in the next couple slides. So allopatric speciation first. Speciation occurs with the evolution of reproductive barriers between isolated population and parent population. Even if two populations should come back into contact at some later time, reproductive barriers will keep them as separate species. So here's our allopatric speciation. We have population divided into two smaller population, but look, they came together and they interbreed, so no speciation. But if we have geographic barrier and we keep it for long enough time, right, and then population come back together, but there is already some isolation between them, reproductive isolation, they don't interbreed, then we have two different species, and voila, that's speciation. We started with one species, we have two species. Sympatric speciation, the one that I told you gonna be explained later. So sympatric speciation occurs in the populations that live in the same geographic area. An example are many domesticated plants, such as oats, potatoes, bananas, peanuts, apples, coffee, wheat. So here's an example of speciation, sympatric speciation in a bread wheat. So that's a wild wheat, this is a wild wheat, and they different species. They might grow in the same territory, right? They can grow close to each other, and occasionally they can even breed, right? They breed with each other. But the problem is one has 28 chromosome, another 14 chromosome. Remember, when they form gametes, egg and sperm, we cut this into half, right? So half of 28, half of 14. When we fertilize them, we have 21, right? So that organism, it can grow, but it's a sterile, right? It cannot reproduce. But what happened at some point of time, doubling of the chromosome. So it's called polyploidy. When egg and sperm was formed without separation of the chromosomes. So egg had 21, sperm had 21, right? And here's our bread wheat with 42 chromosomes. And now it became different species because now it can reproduce, right? Accident during cell division that results in the extra set of chromosome is a common route to sympatric speciation in plants. Of course, that probably never gonna happen in animals, or maybe very, very rare in a very primitive animals, but pretty common in plants. So here we all about these reproductive barriers. And we can have pre-zygotic reproductive barrier and post-zygotic barrier. Pre-zygotic, when zygote never formed, right? So sperm never fertilized an egg. And it can be because, let's say animals reproduce at different time, or they live in a different area, or they have different behavior, or some mechanical isolation. It's just their reproductive organs do not match, or even sperm and egg does not match. So zygote is never formed, so that's different species. Or post-zygotic barrier, zygote can form, right? But the offspring are not viable or they are not fertile. So it can be either they don't reproduce or they have trouble reproducing. Hybrid breakdown means in the second generation they cannot reproduce anymore, right? So when these reproductive barriers are created, we see the evolution of new species. Okay, so, well, after listening all this lecture, you can say, well, what is the big difference? We started with a population of mice, we end up with a population of mice. It might be a different color. Or we start with wheat and we end up with a wheat. It might be a little different. But, evolution from, or differences between a mouse and an elephant is too huge. So how we can explain big changes? So what accounts for dramatic differences between dissimilar groups? And this is the evolution of biological novelty trying to explain us. We have special genes, they called Hox genes, also known as homeotic genes. And they're a group of related genes that controls the body plan of the embryo. So after embryonic segments are formed, these Hox genes and code for Hox proteins, and they determine the type of segments that will form on a given part of the body, right? So, so Hox protein confirm segmental identity, but do not form the actual segments themselves. Well, what it means, like if you look at this fly, right, where the head gonna be, right, where legs gonna be, where wings gonna be, right, or in a human, where eyes will grow. You don't want your eyes to grow on your back, right, or you don't want your arms grow where your legs supposed to grow. That's what those Hox genes partially responsible for, right? Make sure that your body development of your body follows a specific plan. So mutation in homeotic genes can profoundly affect body form, right? So we might believe that a biological novelty, like new parts of the body, maybe they evolved, they appeared because of some mutation within these homeotic genes. So homeotic genes, Hox genes, are very interesting and very fascinating topic, but unfortunately we don't have time to discuss it right now. This is our last slide, and I hope it was helpful.